Hi all. In this video, rather than talk about safety, I'm going to address a couple specific questions that came to me via email. The origin of these questions comes from a website which I show here. It's called truthcomestolight.com, and there's a couple individuals shown in the picture here, Dr. Tom Cowan and Andrew Kaufman, who post a lot of commentary on this website, which is very skeptical of the vaccine and the COVID disease being correlated to the actual virus. So these guys are scientists. They make some coherent arguments, but I'm going to address a couple very specific questions that are posted on this website. Well, I looked at these videos and as a scientist, was a little bit upset with some of the discussion here because a lot of their arguments don't really hold together. They do pose some questions which are good ones as far as where did the virus come from originally, how was it isolated, was it isolated at all, and how do we really know that the virus is the cause of the disease? These are all questions that can be addressed scientifically, and I'll try to answer those with the information that I have. So the first one, does the virus cause the disease? And this is, it kind of strikes me as a no-brainer, but it's worth thinking about. How do we really know? How do we connect those two dots? Here's a cartoon of the coronavirus, or the SARS-CoV-2. You've probably seen something like this before, where there's spike proteins embedded in this membrane, and then there's an RNA molecule inside. So this virus is an RNA virus. But that's just a cartoon drawing, right? How do we really know that this virus is infecting people's airways? Well, here is an electron micrograph of an isolate from the first US case of COVID. So this picture was taken from material that was collected from the nasal swab of a COVID patient. It turns out that in order to get these virus particles to propagate, you have to actually grow them in a dish of human cells. There's a cell culture called vero cells, which are commonly used. And you can get the virus to grow. It's not so simple to just take a nasal swab and then look under the microscope. So there's a couple steps of cell culture that are required for you to see this, but this is one pretty obvious piece of evidence that there are viruses growing in this COVID patient. Furthermore, we have diagnostic testing. There's something called PCR, polymerase chain reaction assays. And these are highly specific and very sensitive tests which detect the presence of a specific sequence, a specific genetic sequence. So for the nasal swab samples that are collected from COVID patients, we can extract RNA. So just for the purposes of explanation here, the green bar in this cartoon would represent RNA extracted from the nasal swab, and the red Lego looking thing is something called a DNA primer, which can be added into the mix, and that primer sequence binds very specifically to a sequence within the virus chromosome. And the way these assays are configured, we can look for the spike protein gene, for example, or any gene that is specific to the virus, the coronavirus. So that PCR reaction then can be carried out, and it's what's called an amplification reaction. So even though you're starting with a tiny amount of viral RNA collected from the nose, or you can collect it from saliva as well if you spit in a tube and extract the RNA, you can carry out this reaction. And it's really the specificity of this annealing event, or binding of this sequence-specific DNA primer, which is diagnostic of the presence of a very specific coronavirus. So to address the question of how do we know that the virus causes the disease, Well, COVID patients test positive for this PCR assay. Individuals that do not have the infection test negative for this PCR reaction. Those are generalizations. We know this assay has a certain failure rate. You can get false positives and false negatives, but generally speaking, this is a very important piece of information to think about. We can track the transmission of the virus between individuals by doing this test. And again, it's a very specific diagnostic for a very specific sequence within the coronavirus RNA genome. So I'm also going to show you this website, which is from McMaster University in Canada. There's a group of scientists that isolated the virus for the purpose of propagating it in a culture dish. So they took nose swabs from COVID patients and they cultured the virus on these vero cells, and they produced electron microscope images like the one I showed you, again connecting the disease to the presence of virus particles in the patient's nose. So I would throw questions back at Dr. Cowan and Dr. Kaufman and say, if COVID does not, if the disease we know as COVID is not caused by this virus, which we can find in these nasal swabs, we can actually propagate it, we can look at it with an electron microscope, we can detect it with PCR. If they dismiss that data, then what is causing this pandemic? Why are so many people dying from what appears to be a viral transmission? To say that this pandemic is not caused by COVID or the SARS-CoV-2 virus, I think is way off base. But the second question that's posed in this link, if they haven't isolated the virus, how can they make a vaccine? This is a more interesting question, and I'll try to address it because it's kind of important to understand in the context of how these new vaccines are made, why we don't have to isolate the virus in order to make a vaccine. So I'm going to go back to this cartoon drawing and remind you that the traditional vaccines are made by propagating these viruses, intact viruses, which include not only the virus spike proteins, but all the other proteins that the viruses express, as well as the RNA in the virus as well. The traditional vaccines require that you mass produce this virus and you purify it away from these vero cells or the human cell cultures that are required to propagate it. Had Dr. Cowan and this John Rappaport guy been criticizing the traditional vaccine technology, this question about isolation is more valid. And I should point out that the traditional vaccines are under development. They're going to be coming maybe in a year or two if the RNA vaccines don't work, or probably even if the RNA vaccines do work, we're going to see different types of vaccines come available. But my point in addressing Dr. Cowan's question, if they haven't isolated the coronavirus, how can they have a vaccine? So the answer to that is, the short answer is that we don't have to isolate to do the new mRNA technology. The virus replication cycle looks something like this. A coronavirus infects a cell, it enters the cytoplasm of the cell, the RNA is released, and then the cell machinery makes new virus from that virus chromosome. So I'm pointing my cursor at the virus RNA chromosome, which is not to scale. It's really a tiny piece of RNA, but it gets turned into new viral proteins, and then entire virus particles emerge from the infected cell. So from one infected cell, you may release, thousands of virus particles. So that's the basis of growing a cell culture which harbors the virus and is used as a bioreactor to make virus, which can be collected and turned into a vaccine. But what if we didn't need to go through this whole life cycle? What if we could just take the viral RNA and just amplify that and mass produce the RNA? Well, that's the basis of the new technology, is to just make viral RNA. In fact, just a piece of the virus chromosome is is made and then encapsulated in these lipid vesicles. And I talked about that technology in the previous video, so you can go back and look at that one if you want. We can avoid the whole propagation in a human cell culture. We can avoid isolating the virus at all, and we can just start with the genetic sequence of the spike protein. So where does that sequence come from? That's actually a fair question, because in order to mass produce the coronavirus RNA to be used for this lipid encapsulation, you have to start with what you know is the real RNA sequence of the coronavirus. So how do we know what the real RNA sequence is? If you followed the reporting on the early stages of the pandemic, the World Health Organization obtained from China the sequencing information on the coronavirus. And they made that information public so that US scientists and scientists around the world could take that gene sequence. In fact, they had the sequence of the entire coronavirus RNA genome. But with that information, it becomes unnecessary to even grow the virus. Nowadays, it's possible to synthesize in a test tube the gene sequence. So if somebody just gives me a piece of paper with a string of A, T, G, C, all of the nucleotide sequences for a gene that's unique to the coronavirus, we can produce that in a test tube without any starting material from the virus or from the patient. So thinking back on it, it must have taken a lot of faith that the information that was being passed on from the WHO and from China was accurate gene sequencing information, because that allowed the scientists to immediately start mass producing the RNA, which would go into the vaccines. So going back to this cartoon, instead of having to assemble this entire virus in a cell culture, what's now being done is to make these lipid nanoparticles, where the RNA component is added entirely from a synthetic source, is mixed together with these 4 different lipids, and these nanoparticles are formed. This is a much simpler structure, and I don't know if you can see that arrow, but that's pointing to the RNA. So the in vitro synthesized and mass-produced RNA is mixed with these 4 different lipid chemicals to make these nanoparticles, instead of growing human cells in a culture dish, taking the virus from a patient's nose or mouth and trying to propagate that, which takes a much longer period of time. That's the old technology. This is the new technology. And again, to answer the question about how can you create a vaccine without having isolated the virus, well, I just explained it to you. This RNA can be made totally in a test tube with, again, some assurance that you know you're working with the right gene sequence, and then the chemical lipids can be assembled or brought into the test tube and mixed together. And there is some art, I guess, into actually making the formulation and making the lipid particles efficiently, but that's beside the point for this discussion. So I hope that serves as a rebuttal to the stuff that's being discussed on that website, Truth Comes Out, or whatever that name of that website was. Those individuals on that website, they obviously know a lot about science, but they're spreading some dubious information. And the obvious point to be made here is the vaccine apparently is working. The videos that I put out previously discuss the efficacy data being 90 to 95% protective. And so if for whatever reason the starting material to make the new technology vaccines was not the perfect sequence, the end result is that we have a vaccine that works and that it is protecting people. So that's it for now. I look forward to putting out more videos here and there, and I do appreciate the questions, and I hope everybody is doing well. I'll talk to you again soon.